Cellid Company Limited, which started from the laboratory in College of Pharmacy at the Seoul National University in 2006, has been at the forefront of developing anti-cancer immunotherapeutic vaccine and infectious disease prevention vaccine. Cellid has accelerated clinical trials for the development of CeliVax, its proprietary technology, and is currently developing COVID-19 preventive vaccines. Cellid achieved the feat of being listed on the COSDAQ in 2019 in recognition of its advanced and innovative technology. In 2020, Cellid built the GMP Center to produce preventive vaccines and cell-based therapeutic vaccines, paving the way for a global pharmaceutical bio company. Cellid is now preparing for a greater leap forward in the new office building in Magok, Seoul, the center of the K-bio industry. It will present a new paradigm in the biotechnology field with the state-of-the-art research facilities and infrastructure. Cellid conducts research on selection and verification of basic candidate biological products and develops the production process for the selected products. Cellid provides a one-stop biosolution from development to production based on the most talented research personnel in Korea. Ultimately, Cellid develops and produces a completely personalized immunotherapeutic vaccine, considering the purpose of use, and manages the vaccine in an optimal condition to ensure the best quality. In particular, the GMP Center, which has introduced systems of strict standards, is equipped with a number of production lines, including the lines for commercial purpose to accelerate clinical development. Based on the annual production of up to 1,000 lots, advanced infrastructure for a simultaneous multi-item manufacturing, and quality testing and licensing services, Cellid is maximizing corporate values through technical partnership and out-licensing to global big pharmas. At the heart of Cellid's vaccine technologies is the adenoviral vector and anti-cancer immunotherapeutic vaccine platform that has been developed with its creative research. Cellid's CeliVax technology has been applied to different cancers containing specific cancer antigens throughout multiple BVAC pipelines. First, the adenovirus vector with tumor antigen gene is delivered into the autologous immune cells. Then, alpha-galactosylceramide is loaded on autologous immune cells. It takes only a day to conduct this process. Now, BVAC for specific cancer patients is injected intravenously into the body. BVAC stimulates natural killer T cells in the body to secrete cytokines and activate natural killer cells. Meanwhile, the adenovirus vector in BVAC produces tumor-specific antigen proteins, which are processed into small multiple immune epitopes. Immune epitopes migrate to MHC1 and MHC2 on the surface of BVAC. Cytotoxic T cells are activated through MHC1 molecules. Helper T cells are activated through MHC2 molecules. Then, helper T cells induce activation of B cells that will secrete tumor antigen-specific antibodies. As such, Cellid's BVAC technology induces both innate immunity, natural killer T cells and natural killer cells, and acquired immunity, cytotoxic T cells, helper T cells, and antibody responses. This maximizes the efficacy of cancer treatment. Cellid's COVID-19 preventive vaccine technology enables a rapid response to infectious diseases through an efficient expression system. First, the gene of COVID-19 spike protein is introduced into the adenovirus vector. Once vaccine is injected into the body, the adenovirus vector containing the gene of spike protein migrates into antigen-presenting cells such as dendritic cells. Then, the adenoviral vector produces a great amount of antigenic protein, and the proteins are processed into multiple immune epitopes. Epitopes bind to the MHC1 and MHC2 molecules on the surface of antigen-presenting cells. MHC1 and MHC2 molecules activate cytotoxic T cells and helper T cells respectively to form memory immune responses. In addition, B cells that recognize antigenic proteins are activated by helper T cells and secrete virus-neutralizing antibodies. In the absence of neutralizing antibodies in the body, it cannot be protected from virus infection. However, when a neutralizing antibody is generated by a vaccine, it binds to the cell binding site of the virus immediately and blocks cell infection. Vaccine-induced cytotoxic T cells also play a critical role in defense against infection. Without virus-specific T cell memory immunity, the spread of infection is facilitated in the body. However, if the T cell memory immunity is established by the vaccine, the T cells can quickly attack the infected cells and suppress the spread of the infection in the body. As the COVID-19 vaccines developed by Cellid are capable of inducing both cytotoxic T cell responses and neutralizing antibodies response, they can effectively respond to viral infections. A new vaccine can be quickly prepared by replacing only the antigen gene to combat the mutated virus variants. Cellid will present a unique and new paradigm in the field of anti-cancer immunotherapy and promote health and a happy life for all mankind. Cellid will challenge to conquer cancer through the CeliVax platform technology and leap forward to become a global pharmaceutical bio company. Cellid.